Welcome back to the deep dive. Today, we're diving into something really cutting edge in cancer immunotherapy. We're talking about CRT cells.
Right, these engineered immune cells, they've been, revolutionary for certain blood cancers.
Absolutely. But there's a big hurdle, isn't there? Solid tumors, they just don't seem to work as well there.
That's the major roadblock, yeah. We see amazing initial responses sometimes, but the T cells either disappear too quickly or they just stop working. They get exhausted or suppressed.
And the main culprit is the neighborhood they move into, the tumor microenvironment, the TME. It sounds pretty nasty.
It really is.
Yeah.
Think of it like a biological fortress designed to keep immune cells out or shut them down if they get in.
So what makes it so tough for our CRT cells specifically?
Well, it's kind of a double whammy.
Mhm.
First, T cells need positive signals to stick around and keep fighting. We call this co-stimulation. They need fuel and encouragement, basically.
Okay.
Second, the TME is absolutely swimming in signals that actively block T cell function, like hitting the brakes hard.
And the CAR T cells we have now, they try to bring their own co-stimulation, but it's not enough.
Often no. The built-in signals just get overwhelmed by the constant stop messages from the tumor environment.
Right, so the tumor is effectively yelling stop all the time, but what if? What if we could somehow rewire the T cell?
How do you mean?
What if it could sense those specific stop signals, things like VEGF or CSF1, which are common in tumors, but instead of stopping, it interprets them as go signals.
Ah, so hijacking the tumor's own weapons.
Exactly, turning a negative into a positive.
That's precisely the breakthrough we're talking about today. They're called TME sensing switch receptors, or T sensors for short.
T sensors, okay, and these are completely new.
Entirely synthetic. They use this concept called allosteric, where binding something at one spot triggers a change somewhere else on the molecule.
Got it. So our mission for this deep dive is to really unpack how they built these things using computers, this de novo bottom-up assembly you mentioned, building from scratch, not just tweaking.
Exactly. And understand how they program these receptors to take those bad signals from the TME and turn them into, well, power-ups for the key T cells.
Okay, let's start with the basic design. If you're building a switch like this from the ground up, what are the essential components?
Yeah, you can break it down into three main parts. Think of it like a sensor system. First, you need the sensor itself. This part sticks out from the cell surface into the TME.
That's the input detector, sensing VEGF or CSF1.
Precisely. Then, on the inside of the cell, you have the responder. This is the part that actually generates the signal, the output, that tells the T cell to activate or persist.
Okay, sensor outside, responder inside, but how do they talk to each other through the cell membrane?
That's the tricky part. And the third piece, the coupling or transmission system. Usually this involves the transmembrane domain, the part that actually spans the cell wall.
Right.
It has to physically transmit the sensor binding the ligand outside across the membrane to activate the responder inside. We call this long-range communication mechanical coupling.
And designing this whole thing, especially getting that coupling right, must be incredibly difficult. You need the ligand binding to make the receptor parts come together or change shape in just the right way.
Oh, absolutely. It requires really precise orchestration. And, when researchers looked at how natural receptors like this work, they found two main ways they get activated.
Okay, what are those?
The first is called the preformed dimer, PFD mode. Imagine the receptor parts are already loosely associated, like two bells touching but not ringing. When the ligand binds, it doesn't pull them closer, but causes an internal twist or shift within the existing structure. It's like flipping an internal switch that makes the bells ring. It's an allosteric change within the dimer.
So a twist, not a push.
Exactly. The second mode is the monomer-dimer equilibrium shift, MDE mode. In this case, the receptor pieces are mostly floating around separately as monomers.
Okay.
The ligand acts like glue. It binds to the monomers and forces them to come together, forming the active dimer structure. That coming together is the activation signal.
So one is twisting an existing pair, the other is forcing a pair to form. Different mechanism.
Very different. And that complexity makes designing new ones hard. Which mode will work? How do you control it?
So how did the T-sensor team get around this? Did they just try thousands of combinations?
They did something much smarter. They used what they call a positive design strategy. Instead of worrying about all the ways the receptor could be inactive or misfold.
Which must be countless ways.
Right. They focused all their computational power on designing the ideal active state. What's the absolute best structure when it is turned on?
That makes a lot of sense. Focus on the on switch working perfectly rather than mapping out all the ways it could be off.
Precisely. They use computational tools like AlphaFold 2 for predicting structure and Rosetta for scoring to find designs that maximize two key things.
Which are?
One, dimerization propensity. Basically, how strongly do the parts want to come together when the ligand is there?
Like turning up the attraction.
Yeah. And two, mechanical coupling. How efficiently does that binding event on the outside get transmitted to the inside?
The signal clarity, basically.
Exactly. By optimizing for these two properties computationally, they could screen tons of potential designs in silico before even starting experiments in the lab. It saves enormous amounts of time and effort.
That computational screening is really the game changer here. But wait, if you're only designing for the active state, how do you stop the receptor from just turning itself on all the time? Wouldn't that wear out the T cell?
That's a great question. And that's where tuning the coupling score becomes critical. It's not just about making coupling strong, it's about making it appropriately strong.
Meaning?
If you design for very strong coupling, the receptor is essentially locked tight in the off state unless the ligand binds really strongly. It resists spontaneous activation.
Okay, so high coupling means low background noise.
Right, but if you design for weaker coupling, you allow for a little bit of leakiness, some baseline activity, even without the ligand. And sometimes that's exactly what you want.
Interesting. So you can actually program the level of background activity.
That's the core idea.
Okay, let's get specific. They targeted two factors found in the TME, VEGFA and CSF1. Why those two?
Good choices, because they're common in many spolid tumors and known to be immunosuppressive or support tumor growth. VEGFA helps tumors build blood vessels, and CSF1 supports tumor-associated macrophages, which often dampen immune responses.
So they're turning villains into informants, essentially. And what about the output signal inside the cell? What did they choose for the responder part?
They used the signaling domain from a receptor called CMPL.
Why CMPL?
Because CMPL activation triggers a whole cascade of downstream pathways, things like JAK-STAT, PI3, CACTMTOR, MAPKIR. Don't worry about the acronyms.
Okay,
The important thing is these pathways are like the T cell's internal power grid and life support system. They tell the T cell to proliferate, survive longer, and maintain its metabolic fitness so it doesn't just burn out at the tumor site.
Got it. So input, VEGFA or CSF1. Output, CMPL pathways for T cell persistence and power. Now, you mentioned programming the activity. How did they do that differently for VEGF versus CSF1?
They designed two distinct T sensors with different goals. The first was the VMR, which stands for VEGFR2 MPL receptor. It senses VEGF.
Okay.
For VMR, the goal was to make it highly VEGF dependent. They wanted very little activity without VEGF and a strong boost when VEGF was present. So they designed it for high coupling.
Locking it off unless VEGF unlocks it, like an emergency booster only deployed in the thick of the TME.
Exactly. Minimal wasted energy. The second receptor was CMR for CSF1, our MPL receptor sensing CSF1.
And the goal here was different.
Yes. For CMR, they wanted some low-level constitutive activity, kind of background hum, even without CSF1, but which could still be boosted by CSF1.
Yeah.
So they designed this one for lower communication or lower coupling.
So the weaker coupling allows that baseline hum to keep the T cells generally fit, even outside the immediate tumor zone.
Precisely. It provides a maintenance signal for persistence, which is really important for long-term control, especially in diseases like myeloma.
This ability to dial in the basal activity versus the inducible activity just by computationally tuning the structure, that's really elegant.
It is, and the experimental results backed it up beautifully. They made several variants of each VMR and CMR with slightly different coupling scores predicted by the computer.
And did the predictions hold up?
They did. For example, the VMR variant, called VMRFL, which had the highest predicted coupling, showed almost zero activity without VEGF, but strong activation with VEGF. And tests confirmed it existed as monomers until VEGF was added, forcing dimerization.
Confirming the MDE mechanism, the glue model.
Right. And crucially, when they plotted the measured activity change, induced minus baseline, against the calculated coupling score for all the variants.
Let me guess, a straight line.
Pretty much a linear relationship. It showed the computational models were accurately predicting the real-world behavior of these synthetic receptors.
Wow, that's validation. And did they measure how sensitive these final versions were?
Yes. The VMRFL needed about 144 picograms per milliliter of VEGF for half-maximal activation. The CMRFL needed around 315 picograms per milliliter of CSF1.
Those sound like tiny amounts.
They are. It means these receptors are very sensitive, capable of detecting even low levels of these factors in the TME, which is exactly what you need.
Okay, proof of principle in the lab is one thing. Did this actually improve cancer killing in living systems in vivo?
That's the million-dollar question, right?
Yeah.
They tested it in two pretty tough mouse models. One was a metastatic lung cancer model representing a solid tumor challenge,
Right.
and the other was a multiple myeloma model, a blood cancer, but one that's notoriously difficult for TARC persistence.
Let's take a lung cancer first. What did they do?
They used a CAR targeting a protein called EVA2, often found on lung cancer cells. They gave mice T cells with either just the CAR or the CAR plus the VMRFL T sensor. And this model was engineered so the tumors produce high levels of VEGF.
Setting the stage for VMRFL to work. And the results?
A really significant difference. The T cells that had both the CRR and the VMRFL showed much stronger anti-tumor activity. They controlled the cancer better, and the mice lived significantly longer compared to those getting just the CRT cells.
So the VEGF sensing booster worked in a solid tumor, but they did a crucial control experiment too, didn't they? Using a version of CMPL that was just always on.
Yes, exactly. They used a known mutant, CMPLW55L, which makes the CMPL pathway constantly active, providing nonstop co-stimulation regardless of VEGF levels.
You'd instinctively think maybe constant stimulation is good, right? Keep the T cells hyped up all the time.
You might think so. But those T cells, CIR plus the always-on CMPL mutant, perform no better than the T cells with the CIR alone.
No better at all. That's fascinating. So the always-on switch actually failed.
It did. It strongly suggests that the context-dependent activation provided by VMRFL turning on the boost only when high VEGF is present in the tumor is far superior. Constant non-specific stimulation probably just leads to T cell exhaustion over time.
It really highlights the intelligence of the T-sensor design. The boost needs to be targeted, not just constant. Okay, what about the multiple myeloma model with the CMRFL receptor?
Right, so CMRFL was designed for that low baseline activity plus CSF1 inducibility aimed at enhancing persistence. They tested CAR T cells targeting myeloma, again either alone or with CMRFL co-expressed.
And these models had CSF1 present.
Yes, they used models where either the tumor cells themselves made CSF1 or the mice were engineered to produce human CSF1 systemically. So the T cells would encounter CSF1.
And did CMRFL help?
It absolutely did. In these CSF1-rich environments, the CAR T cells expressing CMRFL showed significantly better tumor control and extended survival compared to CAR T alone.
So the combination of the baseline persistence signal from CMRFL's low coupling plus the extra boost from sensing CSF1 in the TME really made a difference.
Exactly. It demonstrates that the CMRFL design, providing that gentle background support plus inducible activation, improved the T cell's ability to stick around and keep fighting the myeloma.
Taken together, these results seem like really strong evidence for the whole T-sensor concept. You can rationally design a receptor to sense a specific TME cue and deliver a tailored co-stimulatory response that overcomes the local suppression.
It really validates the whole approach, taking the TME's own signals and turning them from a liability into an asset for the Hayer T cells.
So, wrapping this up, the big takeaway seems to be that this computational design platform isn't just theoretical. It actually works to build these novel synthetic receptors.
It's a major leap, moving from kind of guessing and tweaking existing parts to truly engineering complex biological switches from the bottom up based on predictable biophysical principles.
And the key was focusing on tuning those two core properties, dimerization propensity and mechanical coupling.
Exactly. By modulating those, they could create T sensors with programmable activity, either tightly controlled and inducible like VMR, or with a baseline hum plus inducibility like CMR.
Turning hostile signals like VEGFA and CSF1 into beneficial co-stimulation, directly counteracting the TME's suppressive effects.
Right. And the implications go way beyond just these two examples.
How so?
Well, the design principles themselves, tuning, dimerization, and coupling, they aren't tied to VEGF or CSF1 or even CMPL specifically. They're scaffold agnostic.
Meaning you could swap out the parts?
Precisely. You could potentially design a T sensor to recognize almost any cue in an environment, other soluble factors, proteins on other cells, maybe even metabolites like lactate.
Wow.
And link that input to virtually any desired cellular output. Not just T-cell co-stimulation, but maybe differentiation signals, safety switches, metabolic reprogramming. The possibilities are huge.
So this isn't just about improving CAR T for cancer, although that's massive. It's a foundational technology for synthetic biology generally.
I think so. It gives us a powerful new toolkit for programming cells to sense complex environments and respond in sophisticated, predefined ways. Think about applications in regenerative medicine autoimmune diseases.
That actually leads perfectly into our final thought for the listener. We've seen how they flipped immunosuppressive TME factors into T-cell activators.
Turning lemons into lemonade, biologically speaking.
Right. So thinking broader, what other inhibitory signals, maybe ones common in chronic inflammation or autoimmune conditions where the immune system is mistakenly overactive, could potentially be targeted by this approach? Could you design a receptor that senses the drivers of that chronic inflammation, but instead of activating, it triggers a calming or self-regulating pathway within the immune cell itself?
Using the disease signal to induce self-correction. That's a fascinating idea. Retraining rogue immune cells using their own aberrant signals as the trigger.
It feels like the kind of targeted, context-aware therapy this technology might unlock down the road. Something definitely to think about.
Absolutely. The potential for programmable cell therapies is really just beginning to unfold.
Indeed. We'll leave it there for this deep dive. Thanks for listening.